Hi everyone. In today's video, I wanted to go through the different tests that exist in order to not only identify the presence of bacteria, but also to identify the different types of bacteria that may be present in a sample. Now, there are 5 different tests that I wanted to go through today. The first test is called the indole test. Now, this test is quite important because it firstly allows for the identification in bacteria of the particular molecule called tryptophan, and this is done by a special enzyme, by adding a special enzyme called tryptophanase. Now, this enzyme is basically added to the sample of bacteria, and if there is a color change in that the bacteria turns blue, then it'll essentially mean that the bacterial sample contains the molecule tryptophan. And this tryptophan molecule can actually be used for many different processes. It can form different molecules within the bacterial cell, including indole and ammonia. And therefore, it can have various pathogenic effects. The next test that I wanted to talk about is called the coagulase test. Now, as the name suggests, the coagulase test is used to check if the bacteria has any clotting factors. So say, for example, we have a particular beaker filled with blood plasma, and we have a sample of the bacteria present within the beaker. If the coagulase test is done, then it should be determined that the bacteria are able to clot the plasma. If this particular characteristic exists, then the coagulase test is successful. Next, I wanted to talk about the catalase test. This is quite an important test in the body, as hydrogen peroxide is an enzyme that is used widely by various leukocytes in order to incite a particular immune response. Basically, what I mean by that is that hydrogen peroxide, which is H2O2, is used by immune cells in the body in order to act as a basic method of defense. As hydrogen peroxide is a very toxic molecule, it is essentially able to allow for the breakdown of various bacteria within the body. However, if adding hydrogen peroxide to a particular sample of bacteria allows for the formation of 2 water molecules and an oxygen molecule, essentially meaning that the hydrogen peroxide is broken down into water and oxygen, then the bacteria will be immune to certain immune defense strategies. Therefore, it will be immune to those particular leukocytes. The next test that I wanted to talk about is the disk diffusion sensitivity test, which I'll simply write as the disk test. This test is used with various antibiotics and is not necessarily specifically involved with the bacteria itself, but rather the effectiveness of the antibiotics. Essentially, there can be 2 main different results that can result from this test. Firstly, if we have a disk present here, and say, for example, we have a sample of bacteria that are spread across this disk, as so. Now, say we also have a particular antibiotic that we want to test. The disk diffusion test basically sees is that if we add a sample of the antibiotics in the middle, If the bacteria are able to be affected by that particular antibiotic, then the disc or the circumference of the effect of this antibiotics must become larger over time. Therefore, the larger the disc, the more effective the antibiotics is. However, on the other hand, if we have another sample here with a different type of bacteria present, and say, for example, that this bacteria is highly resistant to a particular antibiotic, then the circumference of this antibiotic will not grow. This is because the bacteria are resistant to it, and therefore the disc diffusion test will be unsuccessful. The final test that I wanted to go through today is called the mannitol salt agar test. This test is actually responsible for checking between the different types of bacteria that may be present within a sample. So if we have a petri dish here, and let's just say that on one side we have Staphylococcus bacteria, and on the other we have Streptococcus. The mannitol salt agar test looks at how well these two types of bacteria are able to survive in a highly salty environment. Therefore, this test essentially allows for the preferred proliferation of a particular type of bacteria. For example, gram-positive bacteria are able to proliferate and live quite well actually in highly salty environments. Therefore, if the bacteria is able to live quite well, then the sample becomes increasingly yellow. This is the case with Staphylococcus bacteria. On the other hand, if the bacteria is unable to live quite well within the sample, then it will become less yellow, and that is the case with Streptococcus bacteria. Therefore, in review, there are 5 main bacterial tests that exist. The indole test, which tests the existence of tryptophan. The coagulase test, which tests the ability of the bacteria to clot blood plasma. The catalase test, which looks at the ability of the bacteria to convert hydrogen peroxide into water and oxygen. The disc diffusion sensitivity test, which looks at the effectiveness of a particular antibiotics. And finally, the mannitol salt agar test, which looks at how well a particular bacteria can survive in a highly salty environment. And there are various bacteria which can be tested with these different tests. I hope you've enjoyed the video. Thanks for watching.